So glycolysis and the Krebs cycle, these two things are used to oxidize or to break down glucose. And so all that's going on here in glycolysis and the Krebs cycles is we're taking electrons away from glucose and we're giving them to these electron acceptors. So that's what we're doing in glycolysis and the Krebs cycle. So the first part of cellular respiration is all about breaking down glucose and extracting as many electrons away from it as possible. And then after you complete the oxidation, you're going to be left with carbon dioxide. And so once you finish breaking down glucose in glycolysis in the Krebs cycle, so after this, now we have all of these electron carriers that have these electrons on them. So what's going to happen, these electrons, or electron carriers, are going to try to give these to oxygen. And so when they're going to give them to oxygen, they're going to go down this electron transport chain that we've been talking about. And then there's another process that's going to happen after you get the formation of water as your product. There's going to be a process called chemiosmosis. And that chemiosmosis is using a proton gradient in order to make energy for the cell. And so we're going to go through and we're going to talk about these all in detail. And so by the time you end up finishing oxidative phosphorylation, you're going to end up with a total of about 36 to 38 ATP per molecule of glucose. So we're generating a lot of energy in cellular respiration for just one molecule of glucose. And so we're going to go through and we're going to talk about each one of these in a little bit of detail. Okay, so the first thing we're going to talk about is glycolysis. And again, glycolysis is the breaking of a glucose molecule in half, essentially. And so glycolysis is always going to happen in the cytosol. And glycolysis is another one of those processes that's going to occur in every single living organism. So everything uses glycolysis. So glycolysis is always going to occur in the cytoplasm. And then again, the process in general of glycolysis is to break that glucose in half in order to make pyruvate. And then we're going to release a little bit of energy along the way. After glycolysis, pyruvate is going to move into the mitochondria. And this is where the citric acid cycle is going to occur. And so the citric acid cycle and the Krebs cycle are the exact same thing, by the way. I think on the test it says Krebs cycle, but citric acid cycle, Krebs cycle, exact same thing. Alright, so it's leaving the cytosol once glycolysis occurs. It's going to feed into the citric acid cycle where it's going to be continued to have these electrons stripped away from it. And in the meantime, we're loading up our electron carriers, our NADHs and our FADH2. And then again, we're making a little bit of energy along the way. And then finally, after the Krebs cycle, all of these electron carriers that we created in glycolysis and the Krebs cycle, all of them are going to go to the mitochondrial membrane at that electron transport chain. And so they're going to drop off their electrons. Those electrons are going to move down the chain and they're going to form water. And so this oxidative phosphorylation, these last two steps, this is where it's going to make 90% of the ATP generated in cellular respiration. So you make a little bit in glycolysis and a little bit in the Krebs cycle, but the majority of ATP is going to be made during oxidative phosphorylation. So consider this just kind of an introductory, showing you the big picture, but in not a lot of detail. All right, so when we're talking about making energy, making ATP, there's two different ways you can do it. Now, the way that it's done in glycolysis in the Krebs cycle is something that's called substrate level phosphorylation. And so in substrate level phosphorylation, it's a way that's used to make ATP, but you have an enzyme that has a substrate that's been phosphorylated, all right? And so this is going to be one of those enzymes in the pathway, and that enzyme just happens to have a phosphate group on it. Remember what we said earlier, when you phosphorylate something, it becomes unstable? So this substrate is now unstable, so it's looking to get rid of it, right? It's looking to get rid of the phosphate. So in substrate level phosphorylation, what's going to happen, you're going to have your molecule of ADP, and you're going to take that phosphate from the substrate and give it to the ADP, so that you end up making ATP. It's as simple as that. You're taking a phosphate group away from a substrate, and then you're giving it to ADP. So it's called substrate level phosphorylation. And so this substrate level phosphorylation, this is going to be used in both glycolysis and the Krebs cycle. So substrate level phosphorylation, substrate level phosphorylation. And then the other way to make energy is actually oxidative phosphorylation. But I don't want to get too ahead of ourselves. Just keep in mind we're generating ATP in two different ways. This substrate level phosphorylation, which is going to happen in glycolysis in the Krebs cycle, and then oxidative phosphorylation, which is going to be the way in which we make 90% of that ATP. Questions so far? Y'all hanging in there? All right. Okay, so again, we're starting with glycolysis. And glycolysis is the process of splitting glucose in half. And so this process is going to have an energy releasing phase and an energy consuming phase. And so we're always going to start with 2 molecules of glucose. And we're going to use 2 molecules of ATP. This is the investment phase, so investing energy. After that, you end up creating 4 more ATP. So the net product is going to be 2 ATP molecules. And again, this ATP is being made by this substrate level phosphorylation mechanism. And so at the end of glycolysis, you actually end up with your 2 pyruvates, 2 molecules of water, 2 ATP, and 2 NADHs. So the main products that I want you to know are going to be 2 ATP, 2 NADH, and then 2 pyruvate. So for each one of these processes that we're going to talk about, you know, technically this is a 12-step cycle. It's got 12 different enzymes and 12 different intermediates. It's extremely complicated. And I'm not going to make you memorize that because I'm not mean like that. But for each one of these processes, you need to know what the starting products are, and you need to know what the end products are. So in glycolysis, you're starting with glucose, right? And then after glycolysis has occurred, you're going to end up with 2 molecules of ATP that we created using this substrate level phosphorylation. You're also going to load up 2 of these electron carriers, because the whole point of glycolysis is to start removing those electrons. So once you remove them from glucose, you have to give them to these electron carriers. And then finally, you're also going to generate the 2 pyruvate, which is the immediate result of breaking that glucose molecule in half. Okay, that was glycolysis in like 5 minutes. All right. I guess we'll go ahead and do a review real quick. So what are the 3 stages of cellular respiration? Yeah, go ahead.
The first one is glycolysis, oxidized glucose, and the second one is correct?
Yeah.
Convert the oxidized glucose, and the last one is the oxidation.
Mm-hmm. All right. Yeah, good. So the 3 stages of cellular respiration are glycolysis, the Krebs cycle, and oxidative phosphorylation. All right, why is cellular respiration an exergonic process? Yeah, go ahead.
Because there's ATP present and there's releasing energy.
Yeah, so because it's releasing energy, or it's making energy. Energy is one of the products. All right, that's how we know that it's an exergonic process. Okay, an electron donor is called the blank agent, while the electron acceptor is called the blank agent. So I know that when you, or not I know, but whenever you oxidize something, you're removing electrons away from it, right? But when you have something that's donating the electron, we call that the reducing agent. Even though it's being oxidized, it's called the reducing agent. And then the oxidative agent is the one that's going to be accepting the electron. So it's kind of switched. But the main thing is I want you to know that an oxidation is donating electrons and a reduction is accepting electrons. Okay. True or false? Carbon dioxide is a byproduct of glycolysis. Do you see any CO2 over there? It's false. And so as we'll learn in the Krebs cycle, remember, is going to complete this breakdown of glucose. And so once, the way you tell that glucose has been totally oxidized is by this carbon dioxide. And so that carbon dioxide is going to be released in the Krebs cycle, which tells you that that glucose has been completely oxidized. All right. Chart the path an electron takes during cellular respiration. All right, I'll just go through these since I know it's new for y'all. All right, so these electrons are starting with glucose, right? And they're going to be taken away and added to oxygen. But we have to make our electron carriers first. So they're going to go from glucose to NADs and FADs. And then from there, they're going to go to the electron transport chain where they're going to meet up with oxygen to make water. So that's the flow of electrons in cellular respiration. We're taking them away from glucose. We're giving them to our little taxi cab drivers who are taking those electrons to the electron transport chain where they'll go down and meet up with oxygen in order to form water. So you're going from glucose to your electron carriers to the electron transport chain to oxygen in order to become water. All right. What is the benefit of the electron transport chain in cellular respiration? Well, this is asking or pertaining to, why do we have to go through all of these little bitty steps? And the reason is because we can harvest energy in a manageable way. So that's the purpose of the electron transport chain, is to help, because you're going to be releasing energy, you just want to be able to release energy that you can control. And so the electron transport chain is going to do that. All right, describe two ways in which ATP is produced. The first way is going to be this substrate level phosphorylation. And then the second way is going to be oxidative phosphorylation, which we haven't talked about, but we'll get to that on Thursday. And then finally, what is the net energy output as a result of glycolysis? So it's basically asking how many energy molecules are you making? You're making 2. So glycolysis is always going to produce 2 ATP, 2 pyruvate, and 2 NADH. And then when we go on to the Krebs cycle, again, we're going to finish that breakdown of glucose, but we're going to add a couple of more electrons to these electron carriers. And then after these first 2 steps of glycolysis in the Krebs, once you've loaded up all those electron carriers, it's going to take them to the electron transport chain, drop them off, let them go down, and meet up with oxygen in order to form water. All right, questions?
What was number 8 again?
Number 8, 2 ways in which ATP is produced. The substrate level phosphorylation, and then the other one is oxidative phosphorylation. So we've already talked about substrate level, and we'll talk about the other one on Thursday. Any other questions?
7.
What is
To
the
harvest energy and what?
in a manageable fashion? So if you just take those electrons immediately away from glucose and just give them to oxygen, you'll have all this energy created. And so you want to, the key is you want to be able to collect it slowly instead of just having it, kind of blow up in your face. Yeah, 4. Number 4. An electron donor is called the, let's see, electron donor, I always get these confused too. The electron donor is called the reducing agent, and the electron acceptor is called the oxidizing agent. Right? Is that, I shouldn't say that in your notes.
Reduction is accepted.
Yeah,
Oxidation is donated.
Yeah, so if you're, oxidation is definitely donating, and reduction is definitely accepting.
So flip-flops.
Yeah, it's flip-flop though. When you're talking about the agent oxidizing or reducing, it's the opposite of what you would expect it to be.
So reduction is.
So in a reduction, you're taking something and you're adding electrons to it. And so the product or the electron donor, let's see, let's make this the donor, and then let's make this the acceptor. So this, when it gives up its electron, it's oxidized. But the donor itself is called the reducing agent. Whereas this is going to be reduced, right? It's going to be reduced because it's going to gain that electron. And so we call this the oxidizing agent. I know it makes no sense. It's backwards. But whatever it is in your notes, which I'm pretty sure that's the way it is, that's accurate. Don't ask me why it's that way. I know it's complicated. But like I said, the main point, I don't want you to worry too much about oxidizing agent versus reducing agent. Just know what an oxidation is and know what a reduction is. All right, other questions? Okay, we'll wrap it up there and we'll finish up on Thursday.